Another thing that you guys took a look at in lab is how different pHs affect the activity of the enzyme. And so to figure out what's going on here, it'll again help to know that in real life, enzymes aren't a Pac-Man looking structure like this. Instead, they're proteins that are comprised of a big string of amino acids that have folded in a particular way. It turns out that different pHs can affect how a protein folds, right? And the reason why is because, well, if you consider, let's just unravel this particular protein for a second so that we see that, again, it's just a string of amino acids. What naturally causes a string of amino acids to fold in a particular way, for example, like this, is the fact that different amino acids have different R groups. So for example, some amino acids, like you saw in class and in lab, some amino acids have an R group that happen to have a negative charge on them, while other amino acids have an R group that happen to have a positive charge on them. All right, so keep in mind that all of these amino acids in the string have some sort of R group on them, but we're just going to focus on this one and this one to keep things kind of simple. Because different amino acids have different R groups that have different chemical properties, that's what causes proteins to end up folding in a particular way. So for example, you know that negative and positive charges attract one another. And so because that occurs, because they attract, that's one thing then that could influence how a particular protein folds. Other things that affect the folding, like you might have mentioned in lab or in class, there are some amino acids that are hydrophilic. So that might cause a string of amino acids to fold so that those hydrophilic portions are facing outside, right, so that they're facing the water. Some R groups are hydrophobic, so that might cause a particular string of amino acids to fold so that those hydrophobic portions are hidden from the water. Right? So there's all kinds of R groups that affect how a particular protein folds. If we're going to just focus in then on this particular example, how would placing this particular protein in, for example, a really acidic environment affect its ability to fold properly? Remember that if an environment is really acidic, what that implies is that there's a whole bunch of free protons floating around. And so then the thing to think about is this. Will this negative portion of the molecule be as likely to bind this positive portion of the molecule if this negative basically has now a whole bunch of other choices as far as positive things to bind to? Right? So if something is really acidic, think about how or if that would affect the ability of this protein to fold in its normal way. Same thing with putting proteins in really basic environments. Right? So if something is really basic, one of the things that characterizes a basic solution, for example, is that there's an overabundance of OH minuses floating around. So same thing to think about, right? If a protein is placed in an environment that has a whole bunch of OH minuses, how would that affect this positive charge's likelihood of binding to this negative charge when it has a whole bunch of other negative charges, basically as options to bind to? Another thing that you looked at in the lab was how increasing or decreasing the concentration of substrate affects reaction rate. One way to define a reaction rate is it's the amount of product that's produced in a particular period of time. So let's, to illustrate things, let's just pick a period of time, 10 seconds. So here we'll define reaction rate as the amount of product that's produced in 10 seconds. If you consider a reaction that involves one single enzyme molecule and one single substrate molecule, it might be reasonable to think that this enzyme can convert that one substrate into product within a 10-second period. So in this particular case, the reaction rate would be one product produced per 10 seconds. What if you end up increasing the amount of substrate that you start with to, say, 3? How would that affect reaction rate? Well, it might make sense that if now you have 3 substrate molecules, that more product would be produced within that same amount of time. Right? So in this particular case, the reaction rate now is 3 products produced per 10 seconds. Right? So it kind of makes sense that if you increase the amount of substrate available, that'll increase the amount of product that's produced in a particular period of time. Right? So same thing, say that you up the amount of substrate even more to 8. It might be reasonable to think that this enzyme can turn all 8 of those substrates into products in a 10-second period. So in this case now the reaction rate is 8 products produced per 10 seconds. So generally speaking, when you increase the amount of substrate, it should increase the reaction rate. There's going to come a point though where this enzyme is basically going to be working the fastest that it can. So in other words, as soon as it converts a substrate to product and that product leaves, immediately another substrate is going to come into that active site to be worked on. So when that's the case, when you hit that point where the enzyme is working as fast as it can, it doesn't matter if you add even more substrate. It's not going to be able to produce any more product within that 10-second period. Right? So say, for example, you totally jack up the amount of substrate. This enzyme can only work so fast, right? So even if you jack up the amount of substrate, there's going to come a point where it can still only make, for example, 8 products within 10 seconds. So whenever that point is reached, when the enzyme is working as fast as it can, so fast that even if you add more substrate, the reaction rate's not going to increase, that's called the saturation point of that enzyme. Right? So again, bottom line, generally speaking, if you add more substrate, that should increase reaction rate, but only up to a point when saturation is reached. At that point, even if you do add more substrate, reaction rate is going to stay the same.